All right, so I'm going to talk about about what we call population genetics in complex ecological communities, and I'll just start at the outset by stating that one of the big challenges of the field that we're going to try and address today is to understand how ecology and evolution work sort of together. Now, traditionally, people have studied the processes of ecology and evolution separately. There's a growing understanding that there are many physical and biological situations in which they work at very similar timescales. And so we cannot ignore one when we think about the other. And so today, what I'm going to try and show you is our first steps in adding ecology to evolution. To do that, I need to tell you a little bit about the classical evolutionary paradigm. One of these paradigms is called population genetics. And one classic question that you often ask in this is that you have a population of many different individuals, which are all the same for now, which I'll call wild type or WT. And at some point arises a mutant in this population. And a question that you might ask is that if you wait a very long time, what is the fate of this mutant? And really, there are typically two answers. One is that if you wait for a very long time, this mutant will go extinct, and all you will see in this population are just the wild type as you had before. The other is that if you wait for a long time, the mutant fixes in the population. Instead, the wild type is gone. And so the question is really, what is the relative probability of this process at the sort of bottom versus the top? And typically, what people have done traditionally for a long time in this field is to acknowledge the fact that these mutants have stochastic dynamics. So if you track the frequency of any particular mutant, which is given by F, that's the fraction of individuals in the population that are the mutant. There are really two parts that determine the dynamics. One is a deterministic part, which is set by selection, a selection coefficient S, and the other is a stochastic part, which consists of birth and death noise, which is determined by a parameter n, which sets the population size. And typically, what you're trying to do is you're trying to look at the frequency of a mutant with time. The mutant typically starts here at very low frequency at very early times. There are many different stochastic realizations of these trajectories. And what you're asking is, what fraction of trajectories reach up at the top at a frequency of 1 and fix? And the classic answer to this has been given by what is called the Kimura formula. So you can analytically compute the probability that the mutant fixes in terms of S and N in the initial frequency. And if you plot the fixation probability P fix as a function of the mutant's fitness benefit S, negative means the mutant is worse than the parent, positive means it's better than the parent, you see a curve that looks something like this. It's very easy to understand this curve. When the mutant is what is called deleterious, that is when it grows less well than the parent, then of course the fixation probability is very low. As the mutant gets more and more fit compared to the parent, the fixation probability doesn't shoot up to 1, but gradually rises roughly sort of linearly with S. And then eventually, of course, when the mutant becomes very fit compared to the parent, it's almost certain that this mutant will fix. So the fixation probability goes to 1. But I want to highlight to you that even though this has been sort of dogma in the field for a long time, it assumes, this picture assumes, and you can actually derive this particular Kimura equation, assuming that you have an environment where there's only the mutant and the parent. This is very different from reality, which we see around us, which is that typically evolution happens in diverse communities. We're all embedded in them. And so reality is that this mutant and parent will typically be embedded in a community of many other species. And the dogma in the field has somewhat been that the Kimura equation somehow still is a good description of what happens in this scenario at the bottom. And so today I'm going to try and show you that if you think of complex ecologies where the number of species in the community is very large, that actually represents a distinct and more universal kind of dynamics. So the way that we're going to do this is that we're going to think about this parent and mutant now embedded in a community. We're going to think of the dynamics of the abundances of each of the species in this community. So that's given by n sub I. And so we're really going to imagine something very general. This n sub I, the abundances of all of the different species, including the mutant and the parent, can change according to some very general function g of I, which represents their growth. This can depend on everything in the community. And it's also going to be given by some birth death noise, which also depends on, of course, how many individuals you have. And so what we have is a universal effective theory of the mutant dynamics embedded in this diverse community. When I say diverse, what I mean is that the number of species in the community is very large. Okay, and what I mean by universal is that this G sub I can be your favorite ecological model. There's very few restrictions on this. And so, for instance, it can be a model as complicated as one that includes the fact that species can interact through resources. So my point here is not to take you through this model and explain it. Just suffice to say that there are many species, all of the species in this community, that are eating resources that can be supplied, depleted, cross-fed, etc., etc. You also model at the same time using the same equations dynamics for the parent and the mutant. And your goal is to predict the dynamics of the frequency of this mutant, which is given by how much of the parent and mutant population is given by the mutant. That's F. So our goal is going to be to compute df/dt. And the way that we will do this is by using dynamical mean field theory. If you do the dynamical mean field theory, and when the dust settles, the main result that you find, the punchline is sort of only one additional parameter is needed to capture the community's effect on the mutant. And so when the dust settles, you get an equation for df/dt, which looks something like this. It actually looks very similar to the Kimura equation with one key change, which is that selection is now frequency dependent. So you have three parameters now instead of two. You still have S, the mutant's fitness advantage. You still have the parent's population size, but you have this new parameter eta, which captures this ecological feedback of the entire community. And so you can now go and study this model with frequency dependence. You can solve the backward Fokker-Planck equation, which is what you use to solve this Kimura formula for the fixation probability. And you can arrive at a similar analytic form for the fixation probability in this very, very general case, which involves some imaginary error functions. But importantly, you can check that in the limit where there's no ecology, when the feedback goes to zero, this formula actually collapses back to the Kimura formula. So in some sense, it's broader and contains what the sort of classical thing contained. And then finally, in my last minute, I'll just tell you one sort of qualitative consequence of this whole picture of having ecology in the fixation dynamics of the mutants, which is that the presence of ecology strongly suppresses the fixation of even beneficial mutants. So to show you that, let's go back and plot this plot that we made a few minutes ago. So we're plotting the fixation probability of a mutant as a function of its fitness benefit. Negative values means the mutant grows slower than the parent. Positive values means it grows faster than the parent. This is what the Kimura formula looked like. I'll now call it no ecology. This is the case where there's no other community present. And you can plot the same expression that I showed you on the last slide for our case. So this would be the fixation probability in the presence of ecology. And you can see that there are some similarities, but importantly, there are some differences that I want to highlight. So of course, when the mutant is much worse than the parent, when there's a deleterious mutation, of course, the fixation probability is still very low, no sort of magic here. But importantly, even when the mutant's fitness benefit is moderately beneficial, that is anywhere between 0.5, you see a strong suppression of the fixation probability. And of course, eventually when the mutant becomes very, very fit, you almost always fit. And importantly, not only do you see a sort of strong suppression, you see an almost switch-like behavior in what used to be a very gradual increase in the fixation probability, which is below this critical fitness benefit, which is given by eta by 2. You almost never fix, and above it, you almost always fix. All right, so I'll just end by saying that the takeaway in some sense is that despite the ecological complexity, I hope you can see that the dynamics of mutants in highly diverse communities can still be captured in a surprisingly simple and predictable way. And this predictability arises because of the high diversity, not despite it. Thanks.
Can you explain why the whole rest of the parameters can be fixed by just one parameter, eta?
Yeah, so really the reason turns out to be that in some sense, the very early time dynamics when the mutant arises are the ones that matter a lot because those are the dynamics where the stochasticity matters the most. You can show that once once the trajectories rise a little bit in frequency, the deterministic parts matter a lot more. So it's because of that it's really the effective competition that you feel at early times that matters, and that's what the eta is capturing. Yeah. I don't think your mic is working.
Yeah. So usually the correlation function and the response of the system are coupled, and you get this consistency condition.
Correct. So in this case, at least the time correlation function that you need to consider to get this is very simple. It's just a delta correlation that we assume, which seems to work very well because the early time dynamics are kind of what matter the most.
when we have this basically ecology, like this interaction without any, I mean, if I don't talk about any fixation,
Yeah.
here also these species compete, and then there is this stability issues there. So
Yeah.
like with that also, I mean, we have like some species might survive, some might not. And now on top of that, when you have this mutation and fixation, it's like, I mean, a bit part of it was like kind of sort of understandable, like the fixation will drop down due to ecology, because I mean,
yeah, so here the question we are interested in is the classical population genetics question of whether the mutant will fix or not, and the question of whether other species will die or survive and whether they'll continue to coexist is a separate one, which you can study, but we don't in this talk.
so combining, and maybe like I will catch you later. And
Sure.
So what is the reason that the selection is frequency dependent, but the noise does not have that frequency dependence?
Because the noise comes from birth and death noise in the actual dynamics.
But that is also selective.
Yeah, so I mean, of course, it's not like the noise doesn't depend on what is happening. It's just that it depends indirectly through what is happening in the interactions. Of course, the noise is being affected by the community, but through the interactions.
But typically in these equations, you get the same term, which is selection and noise have the same behavior, right?
No, you don't.
Okay.
Yeah.
Okay, if there are more questions, you can catch Akshit during lunch.
Thank you.
Thank the speaker again.